So, hello everyone. So, we are going to discuss the last lecture of week 1, that is industrially important product. So far in this module we have discussed this overall bioprocessing, then we discuss how to isolate a potential strain, you got a strains, then we discuss metabolism or metabolic pathway. Further we discuss the nutrients which is required for their growth, maintenance and product formation. So, further today we are going to highlight this a broader scope of bioprocess engineering which enables the production of product from different category. So, we will be discussing some of them today like healthcare product, enzymes, food additives, microbial biomass production and fermented food. So, bioprocessing, as you are going to use these biological agents, maybe microbes, plants and animal cells or enzymes, they will convert the substrate into the product, and this process has been explored for the production of a broader range of metabolites like pharmaceutical and therapeutics, personal care and cosmetics, food and beverage, laundry and leather industries, waste management. So, you can see antibiotics, enzymes, food additives, bioplastics, human hormones and single cell protein. So, I have listed a few examples: penicillin, streptomycin as antibiotics, protease, lipase, amylase, polymerase as enzymes, citric acid, vitamins as food additives, polyhydroxy alkonate PHA and polyhydroxybutyrate PHP and polylactic acid as bioplastics, human hormones, insulin, human growth factor, erythropoietin and single cell protein also like Candida utilis, spirulina and chlorella species. So, in the healthcare we are going to quickly see these antibiotics, alkaloids, steroids, toxins, vaccines and certain enzyme and probiotics. Antibiotics, they are secondary metabolites produced by filamentous fungi and bacteria, is used as antimicrobial chemotherapy, but prolonged use of antibiotics results in the development of multi-drug resistance. Vaccines, they may contain the whole culture of bacteria, it may be the live vaccine or inactivated, and it provides the acquired immunity against infections. The production requires control operation condition and strict adherence to good manufacturing practices. So, for example, if you just see this application of antibiotics, they are commonly used for this skin infection, this UTI, respiratory problem, animal bites, post operative infection. So, this is the most common. When you go for surgery, we are susceptible for the bacterial infection, and after surgery we take antibiotics to prevent infection. Now, antibiotics can be classified based on the mode of action. It is whether it is bactericidal, it means it is killing the bacteria like penicillin, or it is bacteriostatic, it means it is inhibiting the growth of bacteria like tetracycline. Further, it can be classified as broad spectrum, means it is active against both gram positive and gram negative, and narrow spectrum, whether it is active against limited range of bacteria like vancomycin. Further, based on the chemical structure, one can classify like beta-lactam ring, penicillin, macrolides, erythromycin, and peptide antibiotics like bacitracin and nitrobenzene derivative like chlorphenicol. So, how does it works? These antibiotics may inhibit the cell wall synthesis, they may inhibit the protein synthesis, they may inhibit the nucleic acid synthesis. So, for example, penicillin, it inhibits the cell wall synthesis, mupirocin, it inhibits the protein synthesis, and quinolones, it inhibits nucleic acid synthesis. So, all three are essential for the growth and survival of the bacteria. So, by interfering any of them, we end up with killing those pathogens. So, this is the summary we have discussed already. This beta-lactam, polypeptide, macrolides, and this aminoglycides. Here I have listed the producer microorganism as well, like penicillin is produced by Penicillium chrysogenum. This polypeptide antibiotic is produced by Bacillus subtilis. Erythromycin is produced by Saccharopolyspora erythrea, that is a gram positive bacteria. Streptomycin is produced by Streptomyces species, this is a gram negative bacteria, and here the mode of action is again mentioned here. So, these are some more antibiotics which are listed here, like tetracycline, it produced by Streptomyces, this is a broad spectrum, and this is the mode of action, it inhibits the protein synthesis machinery. Vancomycin produced by Streptomyces species, active against gram positive bacteria. Rifamycin, these are active particularly against Mycobacterium species, it hinders in RNA synthesis. Further, there are some antifungal antibiotics as well, so they act against fungus and they hinder the cell membrane synthesis. In addition, I have listed few types of vaccines like live attenuated bacterial vaccine, inactivated bacterial vaccine, cell wall antigens, toxoid antigens, and recombinant vaccines, and these are the causative organisms I have already listed here, and some common diseases like Salmonella typhi causes typhoid, Plasmodium vivax causes malaria. Next common this product is enzymes. It has a vast application. So, enzymes are biological catalysts, mostly they are proteins which lowers the activation energy to enhance the rate of reaction. So, enzyme bind with the substrate, it forms the enzyme substrate complex, and further it releases the product, this is a product and this is your enzyme, this is your substrate. Many times enzyme require the cofactor for their proper activity. So, in that case without cofactor they are called apoenzyme, and after cofactors they are referred as holozymes. And you just see the application of microbial enzyme mostly in the detergent sector, followed by this dairy processes, starch processing, textiles, beverage and brewing, animal feed, bakery application, and some other uses. So, they are applied in a vast area starting from detergent to the beverage, textiles, and dairy processing. So, why enzymes are important? Because they work under relatively mild conditions. as compared to the chemical catalyst like a mild temperature, pH, and in addition the important is they are very much specific. So, there is no production of unwanted byproducts. Further, as this is only going to synthesize your product, so it does not require the extensive separation and purification steps, and being a proteinous nature they are biodegradable and wide range of applications. So, in the next few slides we have listed few common enzyme and their microbial source like protease produced by Aspergillus and Bacillus species, and their application in the duck modification, gluten weakening, even beer brewing, leather batting and tendering. Alkaline proteases they are produced by the Bacillus species. Aspartic proteases they are produced by Rhizomucor species. Chymosin they are produced from the Cluveromyces species. So, this chymosin is used for the cheese manufacturing that we will discuss in the next slide. Aminopeptidase they are produced by Lactobacillus lactis. Alpha amylase mostly produced from Aspergillus and Bacillus species and used for the hydrolysis of starch molecule. Beta amylase produced from Bacillus species and species of Streptomyces. They are used for the production of maltose syrup. Amyloglucosidase mostly produced by Aspergillus niger. Beta galactosidase produced from Bacillus species and this Cluveromyces and Candida fungus species. Glucose isomerase mostly produced from this Arthobacter, Streptomyces and actinoplanes. Glucose oxidase is produced from Aspergillus niger and this Penicillium notatum. So, I have listed few more enzymes here and their producer microorganisms like inulinase from Aspergillus niger, pectinase from Aspergillus niger, cellulase from Trichoderma species, hemicellulose R, that enzyme is xylanase, they are from Cryptococcus species. Now, these are the typical bioprocess. So, we start the synthesis of industrial enzyme. So, we have to see whether this enzyme is intracellular or extracellular. If it is intracellular, then we have to go for the cell breakage, then you have to remove the debris, then you will get the enzyme. And if it is extracellular, we can simply use filtration to separate the cells, can remove the cells, and then you can go for the further liquid enzyme concentrate. You can use addition of stabilizing agent or you can go for its precipitation, filtration, drying, standardization and enzyme preparation. So, one can have this powdered enzyme or one can have this liquid enzyme preparations. So, you need to have this particular microbial strain, so that is starter culture. Then one can use different type of fermentation like solid state fermentation or submerged fermentation, and after the fermentation you have to see whether enzyme is the extracellular or intracellular. So, next product we are going to discuss is food additive and supplements. So, these are the few category like antioxidants, flavor, color, preservative, sweeteners, vitamins, amino acid, organic acid and polysaccharides. So, they are mostly applied as food additive, nutraceuticals and prevention and treatment of certain diseases. For example, amino acids, they are produced by Corynebacterium glutamicum. Commercially available product is glutamic acid. Antimicrobial agent, they are produced by Lactobacillus. Color, they are produced by Blackacea trispora. Flavor, Saccharomyces species. Vitamins, Acetobacter species. One example is ascorbic acid. Stabilizer and thickener, they are produced by Xanthomonas species and commercially available product is Xanthomonas. So, now last one is microbial biomass as a product. So, even you can grow the cell in aerobic condition and whole cell is also considered as product. Like biomass produced during fermentation are also product. So, for this what you have to do is starter culture for fermentation process and inoculum for the food and beverage fermentation. Other purpose is waste treatment process, biopesticide, and in addition, this is very important, they are good source of protein for human and animal feed. In fact, we are going to discuss the single cell protein. So, for this microbial cell grown and harvested for the use as human and animal food. There is dry biomass of microorganisms that are rich in dietary proteins. So, here the advantage is to meet the dietary protein requirement under usual conditions. So, in addition, when you are having the cell that should be rich in proteins and that can be applied as human and animal feed. In fact, they are also called novel food or mini food. So, they are either plant biomass or microbial biomass. So, microorganisms like bacteria, Pseudomonas, Lactobacillus and Bacillus species, they are rich in the protein and used as single cell protein. Fungi, Saccharomyces candida species, yeast, Saccharomyces cerevisiae, candida species, fungi, Aspergillus species, algae, spirulina and chlorella species. So, you just see typically as we have discussed, they are rich in the protein. Protein content is around 30 to 80% of dry cell basis. In addition, it rapid growth of cell and high productivity, they have the ability to utilize wide range of low carbon source to grow and produce this biomass, and this is important because once you are going for microbial process, it is independent of seasonal and climatic variations. So, the product quality is very much consistent. So, you just see they are very much rich in the protein in the range of 30 to 80%. Now, next few slides is already known to us, the fermented food. This is available in our daily household things. The most common is the yogurt. So, use this Lactobacillus species to form this yogurt from the milk. So, most common bacteria which is applied in the fermented food is the lactic acid bacteria. It converts the sugar into lactic acid. Sometimes we also apply this acetic acid bacteria which oxidizes ethanol to acetic acid like Acetobacter and Gluconobacter, and there the product is vinegar manufacturing, and lactic acid bacteria it produces this sauerkraut, pickled vegetable, dairy products. So, in the dairy product you see the cheese, butter, sour cream, yogurt, all are fermented products. In addition, recently we are using this probiotics. They contains live bacteria such as Bacillus acidophilus and Bifidobacterium species to improve the gut health and stabilizes its microflora. So, for example, if we just see the cheese production, the enzyme responsible is the rennin or chymosine. So, this casinogen is converted to casein, which is insoluble, and once it is insoluble it settles down and whey is separated, because milk is rich in the milk protein. In addition, it has some water molecules. So, once you are playing with the protein, you can make the protein from soluble to insoluble, and the same is separated. So, for example, this enzyme chymosin, it cleaves the phenylalanine methionine bond of the casein and eliminates the stabilization of K action. Further, this calcium ion is there in the milk which form the micelles, and thus firm coagulum forms and gets separated from the whey. Butter preparation, for this we use lactobacillus species. In addition, in the fermented food, one common product is the mushroom, where you are able to convert this solid biomass into in the form of mushrooms. They are also good source of protein, and they are mostly produced by non-anexic solid substrate fermentation process. So, here you just see the summary of fermented food product: yogurt, is kombucha tea, vinegar, fermented meat product, and these are the microorganisms which are used, and I have listed the potential applications for these products. So, I will stop here. So, today we discussed the overview of potential of bioprocess engineering from diverse field, starting from this healthcare product, and we ended up with the fermented food. So, today we discussed the potential of bioprocess engineering for a wide range of product. We started with healthcare product, particularly this antibiotics and vaccines. Then we discussed the production of enzymes which have enormous commercial applications, and finally we discussed with the fermented food products, which includes this various milk based products, mushrooms, as well as the single cell protein. Thank you so much.